Hello, Paul Harris.
Hey, Roger. Thanks
How's
for
it going?
very good. Exciting
What are,
time.
what are you up to? What mad inventions have you got behind you there?
Yes, a whole bunch of some new things, which is good to have some fresh stuff here, which is where I'm hoping to go today. Um, yeah, we'll get into a little bit of how Theraphi works with plants. Over the years, we've heard people, tell stories about how they had some dried roses in a pot in the next room, cut flowers, and they had dried out, and they had them sitting in the room next to where their Theraphi was, and all of a sudden they started coming back to life and sprouting and having new buds on them, which is pretty incredible. And we've always talked about Theraphi being able to, project this bioactive field and how it works on all basically life forms. So part of the plant kingdom is what we're going to talk about today, and how the Theraphi interacts with plant kingdom. There's been several groups out there have done some small studies with sprouting and plant growing with Theraphi, and some with Theraphi water, running water through the Theraphi and watering the plants with pretty good success. And so it's always been a back burner project for me to start to do some of these studies, but of course it hasn't been much time. But fortunately, I've got a few young people that got involved in working in the lab with me here, and they put together a little project. First project I put them on was, okay, well, let's see what the Theraphi can do with plants. So we made a little presentation for that today. And tomorrow we'll touch on the energy side of things. So I guess we could just dive right in, eh?
Yeah.
So I'm just going to share a screen here. All right.
Looks good.
Okay.
Yeah.
Yeah, so Theraphi the plant kingdom, I teach a lot of alchemy courses, and we talk about the three kingdoms: plant, animal, and mineral kingdom, and the various components to each of those. And so that's why we gave it the title, how the Theraphi works with plant kingdom. The other thing that's I've talked about a few times in the past is the Aquified as well. So this will be sort of a combination of Theraphi and Aquified experiments that we've just recently started. So these are all preliminary things we've been doing just to get a feel for how to structure future experiments. And we're using both the Theraphi and the water from the Aquified. The Aquified is a system that works on similar principles to the Theraphi, but processes water strictly for agricultural uses. You can't drink this water directly. It's more made for plants. Much like many things like fertilizers and how Leyland's working with the Ormus and that. Some of the best ways to ingest minerals and things like that are let the plants process them first. Sort of a process called coabation, where the plants will absorb the minerals and then people can eat the plants, and then you'll benefit from the minerals because the plants have a way of distributing the minerals throughout the structure of the plant. And when they get dried up or eaten fresh, of course, they're contained in very small nano-bite size, which makes the body and the intestinal flora and bacteria in the body mostly capable of eating them. Then they do that another process of coabation and process them and then provide nutrients to the body. So I think it's an important part for as part of our ongoing projects with the various growing techniques for food and sustainability in the future is going to be a really important topic right now, of course, with shortages and all the train going off the tracks, I guess, within the last year and a half for distribution and all that. So it's a big topic on people's minds. as well as, you know, being able to grow things more locally and to have food distribution more efficient. As part of the problem with the way we've been doing things is we eat stuff, you know, from all around the world out of season, and the shipping and the environmental impact on that. Well, what if it's possible to grow banana trees in Canada halfway up a mountain? So these are sort of the avenues that we're looking at in these processes. And so what we've been looking at so far is germination, the growth cycle, and environmental impact of some of these processes. And we started with just basic seed germination. And these were just seeds that have been treated in the Theraphi. And one of the unique things about both Theraphi and the Aquified, it seems to be that the seed germination rate is very high, like usually 100%. And so we've worked with a few different seeds so far, including peas and beets, radishes, and cannabis. And out of all the seeds that we've done, you know, control groups seem to get about 80%, some less, especially in the cannabis seeds. You might get 50% germination rate out of them. And after they've been run through the Theraphi, and this is just simply a matter of putting them in between the two tubes on the table, we generally notice 99 to 100% germination rate. And so that's really been unique. And so in this first round of the germination studies, the other thing we found is that the germination happens very quickly. Many of these seeds, you know, you'll put in the soil, or even just doing them in petri dishes with moisture. They'll take a few days to germinate, sometimes up to a week or so. In the ground, and they'll take a week to 2 weeks to really pop out. And we've seen some amazing germination results within 15 to 24 hours. And so this opens up a whole new possibility for sprouting seeds. We commonly sprout seeds just in the kitchen and eat them on a regular basis. And, you know, generally they take a week to 2 weeks to get to the stage where you want to be able to get a good hearty sprout for your sandwiches and things like that. And one of the amazing things we realized right away was the incredible growth rate in the seed germination, sometimes within 15 to 24 hours. So as part of the next process, we're going to do some time-lapse videos to show you how quickly they're going. But basically, this first round, we had a germination time between 15 and 24 hours for some of these seeds. They produced their first radical, which was basically when the seed shell starts to open up, and that very first little top root pokes out of the seed. Usually happened within 10 to 15 hours. And we've had plants go straight up to the cotyledon stage, which is like the seed leaves coming out within 24 hours. So they were really strong when we transplanted them. So in this particular batch, we sprouted about 24 hours and then transplanted into soil in 48 hours. And we're working with some snap peas, and in the 24-hour period in the snap peas, the radical popped out and pressed into the soil, and the cotyledon leaves didn't quite come out on the snap peas. The beets as well, in 24 hours, had a good size radical without the leaves. And the radishes had leaves and the stalk forming, but they came out so fast it seemed that they were a little bit weak, and they didn't have enough structure to hold upright, but we planted them anyway. the radishes had the leaves and the stalk. The other thing about the peas, even though the leaves weren't on there, the stalk started really thick. And the cannabis, which usually takes quite a long time, a couple weeks to basically germinate, had the radicle pop out within 24 hours. And so after the plants were placed into the soil, then we basically took the plants and put them in their pots into between the two tubes, right in the center. The tubes were about 7-8 feet apart, and we would do a 9-minute on, 6-minute off cycle. And right away we noticed that even though some of the seeds, the stalks looked a bit thin, they started thickening up really quickly as soon as they got exposed to the Theraphi. And so we ran them through a couple cycles like that each day, twice a day. And the beets formed their leaves and stalks right away and came out of the soil, as well as the cannabis on the second and third day. And so 72 hours in, you can see these are the snap peas and how thick and nice the stalks were. I got a picture on the next slide. So they really grew pretty rapidly, 3 to 6 millimeters. You could see them during the day grow. And by Friday evening, we put them inside of the little propagation dome, and the snap peas really started pushing around the soil. We didn't really, on the next experiment, I'm going to set it up with a clear acrylic side of the pot so we can look at the tap roots coming down and do a time-lapse photo on them, because just to see how amazingly quick it went. So the peas came through really strong with a really thick stalk. And normally when you grow these peas, they get quite tall before they get to put out their little tendrils to grab onto things and grow up as a vine. And this little guy, you see it in the next slide, but he's already got tendrils coming out there and a really thick stalk. And this is 72 hours from basically dampening the seeds. So the beets, leaves and stalks, they pushed up to the soil. The other thing is, when we were running them and watching them, you could almost see them grow while they're running in the Theraphi. They fully emerged from out of the soil on their first Theraphi after being planted. For the beets, the radishes formed their second and third leaves in 72 hours in. The radishes formed their second and third leaves as well, and they were about 3 centimeters above the soil. The cannabis popped out the soil and had its first seed leaves and came up about 1 centimeter on that third day. So here you can see the snap peas here, how nice and thick they are. So here's the original seed leaves down here at the bottom, and already he's got a second node on his stalk and some other leaves and tendrils coming out. And usually peas don't get their tendrils till they actually reach up to something that they can grab onto. They have sort of a real intelligence in their tendrils. It's kind of like the cells at the tips of the tendrils are similar to the cells at the bottom of the roots, where the brains of the plants are. And if you look at some time-lapse photos of some of these plants, you'll see the tendrils will actually look around and they'll look for things to grab onto, and then the plant will grow in that direction. They'll reach out and grab onto it. It's the same with this is why we say the brains of the plants are in the tips of these kind of portions. Same within the roots. Right at the tip of the roots where all the action does, the plants secrete certain enzymes and that, just kind of look at the soil, and they know where to move and where to move towards nutrients. So it's really interesting to see that the plant is that advanced that quickly within 3 days. And these were some of the beets. And the cannabis, we started with clones. And so these are just clippings off of trimmings from plants that were rooted. These ones initially were started with a rooting hormone. And then later on, we did studies in cannabis without the rooting hormones. We'll show in a little bit. And so this is over 2 weeks from their first transplanting out of the rooting stage to 2 weeks later. And so as part of this whole idea of electroculture, I guess you could call it, methods using electricity and growing is called electroculture. And there's been a little bit of work done over the years. Most of it kind of forgotten about or lost, but one of the things that George Lukovsky did was make these rings. So we were experimenting with these copper rings around the plants as well. So he found that he could grow plants and even plants that had growths on them, sort of cancerous kind of tumors or whatever, became healthier when he had these little split rings on them. So we've been experimenting with different copper and creating different wavelengths and coils around the plants as well that we'll have more to talk about in the future. But you can see within 2 weeks, these guys have 3 or 4 branches on them, and they're really doing healthy. The other thing we noticed, what we were doing with the Aquified water, so we're doing a combination of Theraphi treatment, the 9 minutes on, 6 minutes off, twice a day, but we were also putting small amounts of the Aquified water in them as well. We'll show here in a minute. And so the transplanting, so what happens when you transplant seeds or plants, they often become a little bit weak and limp right off the bat. So you definitely have to water them. If you transplant a plant and don't water it right away, they'll often die because they're in shock. And so when we first transplanted the seeds, they normally went into shock, and we found that when we put them in the Theraphi right afterwards, they came out of the shock really quickly. So leaves that would be really droopy, like these guys here, they'd perk up right away, going into the Theraphi. And the same with the Aquified water. At this stage, we could split. We concentrated mostly on the roots, but when we sprayed the plants with the Aquified water, one of the things that we noticed right away is that the leaves perked right up, and they would be really droopy. And we're growing a particular strain of cannabis that really has really droopy leaves, just in its genetics, and being sprayed with the Aquified water sort of gave the plant a charge, and this is before going into the Theraphi, and the leaves were curling up and pointing straight upwards. And one of the growers that we're working with, who's a professional cannabis grower, he was saying, he's never seen this particular strain of plants leaves poke up; they usually hang down. So that was pretty interesting. So in a complete ideal settings for certain plants, there's certain temperatures. Temperatures got to do a lot with it. So the temperature of the soil in ideal settings, you can see, are along here for the amount of days for germinating. And so on our next study, we're going to take some of these longer germination times, 14, 15 days, like parsnips and that. We're going to grow some of these longer germinating ones. Cannabis isn't on here, but it's usually a couple weeks as well. So we've been processing water in the Aquified, and the Aquified is set up with about 80 liters of water in it. So it's quite a large batch. So I would say it's not an ideal concentration. We could definitely go with smaller amounts of water and longer running times to process the water. But we basically ran about a 7 to 10 minute cycle 3 times over the period of a couple hours. Probably within the day, ran each batch of 80 liters, 7 to 10 minutes, 3 times a day. And so the plants got watered with about 12 milliliters of Aquified water just from a syringe and injecting at the roots. And 3 millimeter. Ideally, we found that they liked about 12 milliliters, and this is just mixed with the normal water. So we'd put regular water or control water on it, which is really just tap water. And then ideally, we started around 3 mils of water right at the base of the plant into the roots, but we found about 12 mils was kind of ideal. So it was a very low concentration. So there may be some possibilities of taking a concentrated Aquified water and adding it to a larger tank of water to treat that water. But we'll see. This is sort of just the beginning stages of the processing. So here's how we did and none of these were ideal conditions. We really have been joking about the abuse the plants have gone through. They had gotten some bugs on them, some therps, and they were stressed quite a bit in some of the other plants that we were growing. So the other thing we noticed was stressed plants that were weak and insect infested, and it just had general troubles. Maybe they were neglected by lack of water and dried out. We've been able to restore quite a few of them, almost bringing them back to life. So some of the transplants we were doing, we went away for a few days and nobody was here to water them. So they completely dried up, including the roots. And the plants were almost crispy where you could crush the leaves. And we gave them a little bit of the Aquified water and popped them in the Theraphi, and they came right back and moistened up. And even some of the leaves that were almost dried restored themselves and came back. So none of these were ideal conditions. We're going to set up really good control groups and have better conditions for the next studies. But for germinating the seeds, we basically just moistened some paper towel and stuck them in a petri dish and kept them in the drawer. And at the beginning, we weren't expecting to see any real sprouting for a couple of days, and when we looked at them the next day and realized how far they'd gone, unfortunately we didn't get pictures right at the beginning. So I'm hoping to do that time-lapse photo here on the next round. So these were watered with Aquified water just to moisten the towel and place the seeds in this petri dish, and then they're put in the drawer. So it was the same for the radishes, the snap peas, and the beets. So the same little bit for each, and they weren't given any extra water at all. They just used what was inside of the petri dish because it was sealed. You can kind of drag some of these to see a little bit better. But this is basically within 24 hours. So you can see these radishes here, the seed shells coming off. The radicle is turning into root pretty good, and some of the seed leaves will pop out from inside of the shell there pretty quick. So it was only 3 days for most of the plants to be ready to be transplanted. And that's a major thing for the industry, for food and cannabis, obviously, because to growers, days, the amount of days is really the time in the cycle. The better, shorter you can have those turnaround times in both germination and rooting in when you're doing clones, saves a lot of money. So if you can shave a week off of or two off of your growing cycle in the cannabis industry, you can imagine the amount of savings that would be involved with that. And then in food production, what I'm seeing is maybe a little mini sprouter in everybody's kitchen that uses some of these techniques. And you could just literally be planting. Part of the problems with sprouting is you either grow too much or you're in between cycles, and if you're used to eating sprouts every day, then you kind of have to cycle your sprouts through. But if you know that you can plant and be eating sprouts within 2 days, then it makes it really efficient. And this could change the whole industry for restaurants and things like that use a lot of sprouts and don't have the time or the the equipment involved to grow them. They could be growing these in house, and instead of bringing in sprouts, and they'd be really fresh. So we have a commercial cannabis grower who's agreed to do some experiments with the Aquified water. And one of the things they do is when they make their clones, they take the clippings off these leaves and they soak them in sort of a rooting hormone, and that encourages the plant to spread out more roots. Oftentimes, especially particular strains of cannabis, it's really hard to clone them, and so they use this rooting hormone on a regular basis. So this first batch was just done recently, and so these front ones we tagged, we did a batch with just regular water and a batch with Theraphi water, and then we had some that had no rooting gel on them whatsoever. And the ones, zoom in on here, with that didn't have any hormone rooting and were only treated with the Aquified, were almost as strong a roots as the hormone plants. So again, if we can reduce that hormone in the plants or use a combination of the two, we'll see on their next round doing combinations. Just the Aquified water alone really encouraged the root growth, which was pretty exciting, and exciting for the growers. They were really happy. So far, the roots were also strong too. They didn't break very easily. The roots on the purely Aquified tray versus the rooting hormone tray was about 40%. So even if we had not used the Aquified water, those roots would have barely came out. So. If at all. So it's worth noting that the Aquified control was cloned 3 days. Oh yeah, and those were done afterwards as well. So at this point in time, the Aquified ones were done 3 days later. So 40% of the same roots without the rooting hormone, and clipped and put in their initial medium 3 days later. It was pretty good. We're going to have better control over these experiments. They're out of our control. Some of these ones, we're just kind of relying on the information the growers are sending us. But these are just preliminary tests so we can get the structure set up. And there was some interesting observations. Some growers mix in their plants to, different bacteria and even funguses, and sometimes funguses just show up. I'm not sure if they were purposely growing these particular mushrooms in the bottom of the soil, but some mushrooms appeared in the bottom of this one plant, and So these plants that we did were in a room that was they were kind of waiting for us to get the water to them, so they didn't really do anything to the plants. They didn't spray any chemicals or do anything like that. So they became infested with therps, and they had quite a bit of insects. And we've noticed that when you spray the plants with these therps, they kind of tap onto the plant and they suck the juice out. But the thing about insects and fungi and stuff like that, they're sort of nature's part of the cycle of nature's decomposers. And part of the problem with modern day thinking is that we have to kill these things all off with pesticides and insecticides, herbicides, fungicides, and suicides and genocides. But that nature, they're there for a purpose. They're nature's decomposers, and they'll only pick on the weak plants. So a lot of people have told me in their greenhouses or whatever that if they get some of these therps, they'll always just be in the weakest plant pot, and they kind of live in the soil and suck on the juice of the plant. And then the healthy plants they won't go to. So the idea behind the water and the Aquified being a life-giving force, of course, it's not intended to kill off these other organisms, but what it does is strengthen the plant or whatever you're processing with them, that they become so strong and vital that the insects just ignore them and they move on. And that's exactly what happened in this particular case, where most of the therps, and we've even had some aphids, they would leave the plants that were sprayed with the Aquified water. And in this particular one, a mushroom popped up in here, and I haven't got much more details from the grower. I'm going to have to find out. So no pesticides were used, and often cannabis growers, they'll use some organic pesticides, different essential oils and things like that. And then a lot of growers will actually use real chemical pesticides. And it's part of the problem in the cannabis industry is that you don't know what those growers used on their plants. And so a really big game changer in the whole industry is to have a true organically grown plant that no chemicals have been, touched the plant from beginning to end. And I think that's where we can go with a lot of this. So these plants were never sprayed with anything like that. And the therps just sort of moved on. They jumped from pot to pot, got to the weaker plants, and then the population seemed to die down, and they just lost interest in attacking the plants. So that was about after 2 weeks of spraying the plant, and the plants became obviously more healthy and robust. And now an interesting thing happened in this particular guy, and I'm going to find out more details on this, but a mushroom, a fruiting body of the mushroom, so there's probably some mycelium that was mixed in the soil to help with the mycorrhizal relationship, because funguses and plants work together. Contrary to popular belief, nature's not at war with itself. It is working in harmony. And the fungi under the ground are sort of the communication network, but also the feeders of the plants, and they work together in a synergy. And the mycorrhizal relationship, the tendrils and the intelligent part of the mycelium that are searching forward and moving along in the soil, looking for different minerals, excreting enzymes and breaking things down. They meet up with the plant roots, and this is what we call the mycorrhizal relationship, where they actually sort of embed with each other. And the plants feed the mushrooms sugars that they're producing. So they give the mushrooms, little sugar pills or cupcakes to encourage them to feed them as well. And a lot of excess nutrients that the mycelium is collecting through the soil gets fed directly into the plants. And so these mushrooms here are actually fruited. So when a mushroom reproduces, what we think of as a mushroom as the cap and the stalk is really just the sexual organs of the plant, the fruiting body that's for reproduction, or of the fungus rather. And one of them popped up here, and they sprayed him with the Aquified water, and he went and changed color really quickly. And then I'm not sure what kind of mushroom it was. We're going to have to do some experiments to experiment with different things, but apparently it had a cap on it, and the cap came off of the stem and actually started moving around. They said so. It detached from the stem and started becoming mobile. So the Aquified water has created mobility in the fungi kingdom. Here's a bit closer, and the shots are hard to see, so I'm just going on by what the grower was telling. And so the other thing they did was keep a close eye on the pH of the soil, and one of the concerns was that the pH would change a bit too much with the Aquified water, but that didn't seem to be the case. Everything stayed fairly stable. So this opens up whole new possibilities for doing some experiments with growing plants inside of the with the Theraphi. And a Theraphi could be set up in a large greenhouse, and then so I started thinking, well, seeing how we're working with Tesla technology here, and a big part of Tesla's work was wireless power transmission, and we know we can light fluorescent tubes up wirelessly. Why don't we try and just run the lighting itself from the wireless energy of the Theraphi? So here I just took one of the plant trays and stuck a fluorescent tube on a cardboard box in between the two Theraphi tubes, and this is again about 7 feet apart, the tubes. So as I've shown several times in the past, fluorescent tubes just light by proxy inside of the Theraphi. And so on the next experiment, I'm going to grow a set that are just grown exactly like this, just a wireless power transmission to a full spectrum fluorescent tube that can sit over the plants. And we'll do a study on just lighting with the Theraphi alone. And so the idea is that you should be able to get a combination of the bioactive field of the Theraphi along with the full spectrum light. So we're just adding an extension to the plasma within the tubes themselves. And the beauty of this is, of course, burnt out tubes work just as well as brand new ones. So we could save some environmental impact and utilize older fluorescent tubes. It'd be great to do a study showing plants that have grown up on tubes that were destined for the landfill. There's probably a few carbon credits you could get for that. So that's part of the idea here in the future. Oops, I lost my thing. So the interesting thing, it makes sense that the Theraphi lights up the fluorescent tubes because it's similar to, it's just an evacuated vacuum tube, just as similar as the Theraphi tubes, with gases in there that get excited into plasma. So I was curious to see if there could be enough. They make replacement tubes for these that are full spectrum LED tubes. Because traditionally, these fluorescent tubes have a high voltage ballast. That's that little black box that leaks out that carcinogenic goop inside of the transformer in the ballast inside of these fluorescent tubes. So they've made modern versions using full spectrum LEDs instead of the old style fluorescence to replace these. And of course, you got much more control over your wavelengths of light. So they've adapted vegetative growth stage LEDs and flowering stage LEDs. So the question was, would these LED replacement tubes, because they're designed to operate on the high voltage ballasts of the traditional fluorescence, would they work in the in between the tubes just like the fluorescents do? So here's a zoom in on the, if I can catch it here. So you can see all the individual LEDs, and they're basically just a long array of LEDs that are designed to run on a higher voltage. So there must be some kind of
I think your screen is frozen. Sorry.
Yeah, I paused it here just to show you the LEDs. Sorry.
Mm.
So they must have some kind of system, I don't know exactly how they work, but that takes the higher voltage and makes it a very low voltage, because all these LEDs run on a very low voltage. They might be run in a series or something like that. So here I'm just throwing it on the table.
Oh, we're still seeing the same slide.
Okay, you don't see it now on the table?
No, we just see the.
Yeah, the slides aren't moving through.
Oh, you know what? My share might.
Yeah, maybe exit and restart.
Did you see the last video with the tube on the plant?
No, I don't think so.
Okay. I think that's what it is. I got to switch. Okay, let me go back to this.
Yeah, just restart the share.
Quick time. Okay. All right. Now, do you see a picture there?
Yeah.
Okay. All right. Well, let me go back to the last video then. Okay. Do you see a different one here with the plants on the table?
No.
It's only
Looks like an Indian blanket.
Okay. I guess I got to do this for each individual one that I have here. Sure. Okay. Now you should be able to see it. Okay, so this is the tube in action of the last picture that you saw. I thought it was playing this video a minute ago, sorry. So you see that now where the tube lights up?
Yeah.
Okay. So yeah, this is basically the wireless transmission, where the tube's really just sitting in between the two tubes. There's no wires connected here. And the fluorescent light, this is a full spectrum sort of grow light fluorescent. So yeah, the next study I want to do is just growing the plants under the light that is being lit wirelessly by the Theraphi. And so that's the fluorescent tubes. And here's the LED. So interestingly enough, I thought, well, is it going to light these LED tubes? And so now can you see the LED tube on the angle here?
Yeah.
Okay. So you can see all those little LEDs, and they can be very specific for the wavelengths for growing. So there's two different types, one for vegetative state and one for flowering. So I'm just tuning up the Theraphi here. And as I go closer, you can see all the LEDs are lighting up. So interesting that, you know, it doesn't have to, it's getting enough power just being in the Theraphi to light up the LEDs. And they'll go fully bright. It's just kind of sitting in the middle of the table. And so my body is part of the antenna essentially at this point. That's why it gets brighter with my hand. But if I move a bit closer to the tube, it fully lights up. There we go. Let's zoom in on those guys there. And next video we have so part of what you can really see using the fluorescent tubes is the field effect around the I use the fluorescent tubes to look at the geometry of the field, and when you hold the fluorescent tube, you're seeing this video here with the fluorescent tube lighting. You see that one is running?
Yep.
Okay.
That's running.
So this is at each end, and one of the unique things, you know, I've talked a lot about over the years how where the field converges at the center, the 0 point where power goes out. Here you'll see this 0 point area, or better named counter space, as Eric Dollard's been talking about, and we'll get into a little bit more about that tomorrow. But you can see the field cancels out and it goes to a null point and this dark spot right in the center of the tube. So that's the box, the main coil box is underneath the table there. And you can see it actually dim as it gets towards the center as the two 180-degree out of phase tubes cancel each other out at the convergence point in the center. You can place the tube on the table, and if you adjust things just right, you can actually see the longitudinal waves in the fluorescent tube. So one of the beauties of Tesla coil is that it's the ideal producer of longitudinal waves because there's no metallic core in the center. It's like an air coil. It's really capable of creating a lot of harmonics within the coil. And these harmonics are the perfect way for propagating longitudinal waves. So I'm just trying to get to the next video here. I keep losing my zoom. Okay, share screen. There we go. So one of the things I've talked about this in the past where you can do a really good visualization. Do you see the tube on the table here now?
Yep. See that?
Okay.
Yep.
Okay. So you can actually see the longitudinal waves propagated at really low frequencies. Of course, Theraphi is like a broad spectrum of harmonics from really low frequencies to ultra high. And it really picks it up well in these fluorescent tubes, these standing waves like Roger talked about earlier. And then you can see them move longitudinally, and they'll either, depending on how you look at it, it's one of those things you can see it going both ways, and it depends on the shutter speed of the camera. So this is just the tube laying on the bed in between the tubes. But do you see the moving longitudinal waves within the tube?
Slightly.
Yeah, it's really hard to get. Like when you see it real time, it's quite clear.
Yeah,
See
I've
if I
seen
can
it in real
pause
time.
it. Yeah, but this compression and rarefaction, and they always either converge towards the center or they come out from the center. So from this point of counter space, they'll, in this particular, it depends on the frame rate and everything of the camera, but you can see them. To me, it appears that they're coming away from the center here, but they'll both move towards that center space and away from it. So stop that. And see what else I have. We'll probably go to questions here pretty quick. So yeah, that's kind of where I wanted to wrap up here.
Okay, very good,
Yeah.
Paul. So what sort of radius from a Theraphi tube would you have for lighting up LED lights? And
Yeah, the LED lights seem to be about 10 inches away from the tube. So you could also add on as many as you want, much like with the Quantify, you could put a larger spectrum of them radiating out in a circle around the tubes to cover a larger area. So I kind of visualize a room that would be set up like that, where there'd be the Theraphi and the tubes were maybe twice the length of so 8 to 12 feet apart, twice the length of the 4-foot LED. And then you just do an array of them in sort of a fan shape, like spokes on a wheel around the tubes.
okay,
And now you could experiment with different axes and stuff like that as well. But you could cover a good 20-foot by 20-foot size greenhouse or grow area
Yeah.
for sure. Yeah.
Well, for sprouting, it's usually very concentrated area anyway.
Yeah, and sprouting doesn't need light. Like we sprouted our seeds in those petri dishes in a dark drawer, so that'd be something else to experiment with too.
So ideally, you would have Theraphi spaced out in a growing area,
Yeah.
kind of like every like a grid like every 10 feet or 15
Yeah,
feet.
20 to 40 feet. So if each one spread out, I guess it would be, if they were 10 feet apart, yeah, you would get 20 feet square. So you could have them a different set of Theraphi tubes in every 20-foot square space. So 20 by 20 space.
Well, all we'd need is a control, all we'd need is, say cannabis, a controlled area, and then right next to it, well, a little bit further away.
Another room, yeah, another building, preferably.
Well, yeah,
that was
because
part of the problem we had. Yeah, our sprouting room and stuff was right above the Theraphi, so we had problems with the control group, because we were like, well, the control group is going right through the wall and affecting the control group.
Yep.
So
Yep.
that's part of the plan now, is to make
Field.
2 separate rooms. Yeah, maybe
Yeah.
different buildings.
Yeah, or the other side of the planet.
Yeah.
Interesting, very interesting.
Huh.
And the Aquified, that's very, very interesting. Now, you didn't do any analysis of the water.
No.
Yeah,
Not yet. Yeah, that's to come. I'm playing with a few things. The one thing I know is it really reduces the oxygen reduction potential, and the pH it goes down as well. It becomes more acidic. And
okay.
that's due to the nitrogen in the water.
Hey, Paul, this is Glenn here.
Hey,
Hi.
Glenn. Hi.
Speaking of water, sorry, I had to butt in.
Yeah.
Did you ever try sprouting the seeds in water rather than dry?
No, not yet.
And I'd encourage you to do that. Hate to add another variable, but I'd love to see you try, or me try, or I'll send you some water, structured water versus regular water.
You're right. Yeah.
The structured water will do the same thing, and I'd love to see whether there's any synergism there.
Yeah, synergy, as well as with the imploder, I've been thinking about too.
Or the imploder,
Yeah. And definitely structured water. I think these things are going to work in pretty good synergy, and it may be that they'll amplify each other, because either you're lowering the surface tension, increasing the structure with water already starting, like, so run it through the imploder first, and then into the Aquified, or vice versa. It's going to be interesting to see how they'll complement each other for sure.
Right. Yeah, and if you ever want me to measure conductivity, you can just send me the water after you treat it.
Yeah. Well, I should get you a batch of that here pretty quick. Yeah.
Yeah, we'll have to coordinate. We'll do that later. But yeah, nice chatting.
Yeah. I caught your talk, a little bit of it yesterday, but again, at question time, my phone rang.
Oh, there you go. Right.
That was great. Yeah.
Hey, Paul, are you done? Or
Hey, Stefan.
hey, it's great to see
Still
you.
doing questions.
Yeah, awesome. Yeah, I have two questions. What frequencies are you running through the Theraphi to light up the bulbs?
Yeah, so that's Dan's frequencies that cascade, the golden mean ratio cascade.
Nice.
Yeah,
So you can run multiple frequencies at the same time
yeah,
to power it?
they're all embedded together like a complex waveform.
Nice. And I know, like, when you're recording the plants, there's the Dominator device, and you can actually turn it into music.
Yeah,
A thought would be to amplify the music and put that through the coils.
yeah.
So you're actually speaking plant language back to it.
Real biofeedback.
And you can quantize it to tune it to 432, which is more in harmony with the earth, and use specific scales like a pentatonic scale versus a chromatic scale to give it intelligent chaos. And then you can use tuning forks behind that. Now, has anybody ever tried electrified root systems? Because the water, and then, but does that affect the plant's ability to communicate with each other? Are you like overriding its protocols, or would that be that? Yeah, that's a question.
Yeah, that's a good question. Are you affecting the plants? But definitely that's the whole thing about this electroculture that people have done traditionally is they've put long strings of antennas over top of a row of plants, let's say, and there's two systems, sort of a grounded system and an ungrounded system. So an ungrounded system would just have an antenna to basically pick up the charge from the air and put it across the top of the plants, and they have a natural ground and in the ground. And then the other way they've set it up is to have the antenna up on one side, and then the roots of the plants also really grounded through another copper line or something that runs through the soil as well.
Could you just run two copper pipes and one positive, one negative, and then run the charge through the ground?
Yeah, and so that traditionally they did AC and DC systems and just natural systems from ambient electrostatic energy from the atmosphere. So it's kind of like a lightning rod that channels it through. But then they've also done DC systems with a positive on top and a negative on bottom, and vice versa. And there's some slight differences in DC in your polarity. But some of the AC systems have had just as much results. I might do a talk in the future on sort of the history of electroculture.
That's exciting. Really, really love your presentations. Thank you so much for everything.
Thanks.
Yeah.
Hi, Paul. Sigmund here. Hi.
Hey, Sigmund. Good to see you.
That's good presentation. Yes. Your Tesla coil, have you the same frequency in the both, or you change the frequency and pop you the same frequency in the same?
Right. So no, it's just the standard Theraphi, and they work. You know, the phases are alternating 90 degrees up, 180 degrees out of phase. They're alternating up and down like two buckets of water sloshing up and down.
Yes, that's good. And have you? Is it possible to catch the frequency from the plants in the coil?
Yeah, well, that's what Stefan was just kind of saying there. He's working with plants with those biofeedback, like Cleve Baxter had started doing, and the Dominator and other people have created systems where they actually take the EEG or EKG kind of signal from the plant with, you know, an EKG, highly sensitive amplifiers. And then they take those waves and they convert them into quantized MIDI notes. So then you plug it into any kind of keyboard or synthesizer like that, and you listen to the plant in actual, real quantized notes. So you get the scale of the plant. And the plants can play the music. And so what Stefan was saying, it'd be great to use that as a feedback device and then feed it back into the plants and let them
Yes,
That's
great.
yeah, that's definitely some another back burner project for sure. Gotta come.
I'm doing that for you. I'm actually doing
Okay.
a whole singing plant garden, sonic lumen garden, where I
Yeah.
can do 5 plants at once with surround sound, and then we're gonna harvest the herbs from the plants. And then so you get a multi 5D sensory experience with multi-projection that sound reacts to the plants. And then I have a brainwave interface that converts your brainwaves into music. So the left brain will do one side and the right brain will do the other. You sit in a sound chair and then have the tea of the plants, the smell. And we're going to do like a tarot card deck of all the plants. So the hyssop plant or the bee balm or the, basil, and cumin seed to whatever, so that, yeah, that's coming up soon. And I'm going to start a newsletter, seraphi.org, SERAPHI.org. And the Sarasoma site is on it. That's my vibroacoustic relaxation company that has all the study of the plants and the vitality and all that stuff. So it all relates together.
Oh yeah. Yeah, Stefan's organization there is Theraphi.
Yeah.
When we met in New York, and you came down on that one talk we were doing at Rogers, and we kind of
we both started. I was in San Francisco, and I think we started at about the same time. I was early 2000. I think I started my company, but
yeah,
that's awesome. I'm so elated to be doing it in this life, because I know this is all the technology for. In Atlantis, we lived up to 800 years. And so it was the plasma tube technology that allowed cellular growth to keep sustaining and rejuvenating. But hopefully we get to do a prototype soon. That's
for sure. Well, this is the moment we've been waiting for, isn't it?
Yes, definitely.
Yeah.
That's good. Have you, I have grown plants with electric power, like you say, power, with 20,000 volts hour, and it's growing very fast. But have you tried to combination this therapy with electric power?
A little bit. Nothing that I've documented, but I've set up a few things and set up those Lakovsky rings as well. But I think over the winter here, that's sort of my indoor project. And come springtime, I want to take everything outdoors, and we'll try it out there too.
Yeah. How high power have you used? Have you?
Well, I guess most of the power ranges were low volt, like from the 10,000 volts, I guess, in the high voltage range, up to about 300,000 volts.
But have you still DC, or have you posted the DC?
Just AC. I haven't used DC yet, but that's come up and coming. We should try to compare the difference for sure.
Hello, yes. Can you talk to us about the Quantify? Do you have any new research to share with us about the Quantify?
Yeah, I do. No, I'll touch on the Quantify a bit tomorrow, because I think we're out of time. Right, Raj? Is the next?
Okay.
Yeah, you've got. We can do a couple of more questions, because the next presentation is mine, and it's not. I don't have to go for the whole.
Oh, okay. I was just worried we're cutting into somebody. Okay.
Yeah. No, another 5 minutes. It's fine.
Okay, so yeah, Quantified, same kind of thing. Obviously, it'll do the fluorescent tubes and things like that. So the nice thing about Quantify is you could extend those modules a lot easier and have multiple coils for in those different tube arrays. So setting up a greenhouse, it might be a little more practical to use several smaller coils rather than a couple big coils. One of the things about the Theraphi and running 2 Theraphis together at the same time is they don't kind of get along. It's the same with a lot of Tesla coils. If you run 2 coils at the same frequency, they'll battle it out, and whoever hits the optimum peak usually wins, and it'll just punch out the other coil. So that's the big part about the Quantify, and it was a big hurdle to get those coils to talk together so they can work in tandem. And it might be a little bit tricky to add on to them, but yeah, definitely the quantify could be used in the same kind of situation.
Okay, Paul, we've got some questions in the chat window. So Wanda asks, can you use a sound frequency to ground after Theraphi session in cold, snowy weather when you can't walk around bare feet outside? So can you use a sound frequency to ground after a Theraphi session in cold weather?
Okay, I imagine you could run like Schumann type frequencies through to give you that connection to the earth. The earth's ground is kind of connected to that Schumann frequency. Of course, everywhere you go, you look at those geo maps where they do the radio mapping, and you look at the earth all around, and ground can kind of be seen as counter space as well. If you visualize what we do with electricity and how our power grid is set up, we're utilizing the ground as sort of the sink, and where that power goes, nobody knows. So we're throwing the laws of thermodynamics in here, and so everybody's been searching for the so-called free energy and getting energy for nothing. I mean, that's the hard part. The easy part is to see that the laws of thermodynamics, the second law especially, is totally violated by where does this power go, because it just disappears. So if it can't be created or destroyed, well, yeah. So the earth is sort of like that. You can view the earth as counter space essentially. And when you're in the Theraphi, yeah, that counter space point in the 0 point at the middle is like doing a natural grounding for sure.
There's a question from Ashley. Paul, can you explain again the fluorescent tube demonstrating the different points frequency within the field?
Okay, sorry, let me clarify that question.
Okay.
Sure.
Go for it, Ashley.
Thanks, Roger. So I actually often use that. I have a fluorescent tube, and I often use that as a visual for people who think nothing's going on in the tubes.
Yeah,
right?
Impossible. Yeah.
And it's just like it's just magic trick, right? But so my question is, when you were in your video, when you're like moving the tube around, and I understand that when you back the tube off, the tube fades as the field dissipates, or as as you get out of that bubble, right?
Yeah.
So, but I don't. So my question is, I mean, I understand, like I see the phasing, the inside the tube and all of that, and I see that when I do it myself. But what I'm a little confused about is when you move so in terms of the actual field within the field, how just clarify that a little bit.
Well, that's why I was holding it up vertically, so you could see that. So if you hold it up at one end of the tube straight up and down, and you bring it, it'll fully light at each end. And as you bring it towards the center, you'll see that light shorten as it moves through that center point.
Oh, you mean so the outer part of the tubes get dark? Yeah.
So there's a dark void right at the center, where in a transverse wave, naturally they get canceled out with the phase relationship between the tubes. But it doesn't make sense in a transverse electromagnetic wave. That's where it should be the most concentrated, because it's the closest point between the two tubes. So it's kind of like a reverse thing is happening.
Oh, you mean just visually?
Yeah. Well, no, in reality, or in the field. In the field.
In the field. Okay.
Yeah. So it's the same with the longitudinal waves, how they're both moving towards each other. You'll either see them in the fluorescent tube depending on your eye, depending on the lighting in the room, depending on the shutter speed in the camera, if you can capture it.
Right.
You'll see them either converge towards the center, or they'll be coming out from the center and towards the tubes.
Right. So then does that mean that whether you're putting plants in there or humans in there or dogs or whatever, animals, whatever, that let's just say you're putting a small thing in the very center of the table, is that
Absolutely.
Not the best place to be?
Yeah, absolutely. And your body is sort of this non-linear capacitor. You're like a capacitor in the circuit. So you become part of the circuit. And because you have different densities of tissue from bones to cartilage to, soft flesh, you can picture those same kind of compression and rarefaction longitudinal waves that you're seeing in the tube moving right through your body, because they are, and they're all at different scales. The ones you see in the tube are fairly slow enough that you can see them. So they're a really low frequency within a few hertz, right? But they're in the high frequencies too. So as you go higher frequencies, things get smaller and smaller and smaller. So that's the fractality in the Theraphi, is it has such a broad spectrum range. So every cell in your body, as I've said, is getting sort of a little micro massage by these little pulsations of compression and rarefaction. And that's how we think it's improving that charge on the cell is improving the waste elimination and cellular function and metabolism, basically.
And Paul, you might also say that counter space kind of sweet spot is where the phase conjugate collapse is most optimal, and therefore the translation into longitudinal through the plaque. So there, collapse is perfect, which is what makes it the sweet spot. It's probably the most negentropic place.
Exactly. As Hans explained so many times with the pine cones kissing and everything that they kiss, and everything converges, and the magic happens is right when that wave collapse from a transverse electromagnetic wave collapses, and then it goes longitudinal. So that's why you see those longitudinal waves compress out from that point.
Sweet spot.
So then so the sweet spot, say like when you have your table, like what Paul, it was in your video, like a lot of us have, just that standard massage table.
Yeah.
So we can all assume that the sweet spot is in that center.
Right in the middle. Yeah.
And let's say somebody has, liver cancer or whatever, and they could only sit. They would want to sit right in the center of that table to have that optimal sweet spot on that internal part of their bodies. Let's just say, for example.
Yeah. You could definitely arrange it so their chair was slightly off to get the liver into the center sweet spot.
Right. So that's why if you're going to target the tubes to a certain area of the body, you want to get that center sweet spot right in the middle of the tubes.
Well, the beauty of that, like initially in the beginning, we were putting the tubes real close together and putting your arm in there or whatever it was.
Right.
But then we quickly realized that as you spread the tubes apart, it opens up that envelope, and it's really like a vesica. That's in the center, right?
Right.
And so this is the magic of the whole vesica happening, is the sweet spot in there, and it just really fulfills, everything we've said about nature.
Right.
And so as you widen it up, and if your tubes are wide enough, your whole body's in there. So we kind of gone along with the whole body process being in there rather than trying to isolate certain organs. Because why not just because it's a collective holistic thing that your body's receiving the signals and working the whole body.
Sure, that's a good point. Yeah. So it's best just to do the whole body. I mean.
Yeah.
Yeah. Okay. Thanks.
Great. Thanks, Paul. Brilliant.
All right.